Hello, everyone. Thank you for joining us today for this first of Boyce Thompson Institute's new Breaking Ground discussion series. My name is AJ Bushy. I'm in the communications department here at BTI. And today we are in conversation with Dr. Joyce Van Eck, Associate Professor at BTI, who will be telling us a bit about her research and about her Physalis Improvement Project, which is really, really cool. So hi, Joyce.
Hey, AJ. Hey, everybody.
As we get used to doing everything remotely, that's what we're doing here today.
Yeah.
A couple quick housekeeping announcements. During the session, all participants will be muted. You can ask your questions in the chat box, and then we'll do a Q&A at the very end of mine and Joyce's conversation. So thank you again for joining us today. And so Joyce, let's start with a little bit of background. Can you tell us a bit about yourself and how you came to be at BTI?
Sure. I grew up in Pennsylvania. I got hooked on science when I was in elementary school, especially when the teachers, well, actually nuns, I went to Catholic school, did experiments as part of our science classes, and that really drew me in. I did my undergraduate work at Penn State University. I majored in plant science with an emphasis on plant breeding and crop improvement. I then went on to do a master's degree at the University of Delaware, and then I came to Cornell to do my PhD, which I did in the plant breeding and genetics section. And so it's, I've been involved in crop improvement my whole career. And it's, I have a bunch of hobbies. And one of those is gardening, which isn't surprising.
Makes sense.
Yeah, I got my love of gardening from my parents. I really enjoy picking out the seeds and germinating them. My husband built me a light rack system in the basement, so I germinate all my seeds, and
Wow.
It's fun watching them grow. I think what I like the most about it is that I can give my family fresh vegetables, and I hope I've instilled a love of gardening in my two boys that continues through their lives.
Yeah. How big is your garden? How many crops do you have in there?
Yeah, it's not very big. It's small. It's just enough for us. So I don't have all this surplus that I have to do canning, but I do have enough to give to friends and coworkers. Nice. So kind of the common stuff: lettuce and tomatoes, and interestingly enough, I have a battle with the chipmunks and ground cherries and golden berries. So they get to them before I do, even when they're ripe. So unfortunately, this year I don't have any ground cherries or golden berries in my garden, but I've got other things like potatoes and cucumbers and some kale.
Nice. I started tomatoes from seed for the first time this year. They're doing okay.
Oh, good.
So can you tell us a bit about your lab at Boyce Thompson and what you study?
Sure. So I have 10 people in my group. It includes research technicians, postdocs, an undergraduate, and a new graduate student in the group, Savannah Dale. And I see the slide is coming up here. Savannah's down in the corner there in that inset. So the people in my group are supported on research projects that are funded through the National Science Foundation, the U.S. Department of Agriculture. We also have some personnel who are involved in what's called the Center for Plant Biotechnology Research at BTI. And those people help support the studies of gene function and crop improvement of groups within BTI, at Cornell, and off campus as well. And for the plant species, we work with a number of different plant species, but the ones we work most with are tomato and Physalis, which is the ground cherry and golden berry.
So that's what you do in your own lab, right? Is primarily and Physalis?
Right, it's primarily what we do in my own lab, right?
So for the Center for Biotechnology Research, is that all kinds of species, or is it still pretty much just those two bunches?
It's all different species, but again, it's primarily tomato. Tomato is a great model species for understanding different plant characteristics like plant growth habit and fruit biology. But we also do some what's called model species. There's two grass species that we work with called Soteria viridis and Brachypodium that some groups are using as models instead of growing something like corn, which is, grows really big and it's difficult to grow, and especially in a greenhouse. So by using these model species, it really helps them understand the gene function a lot easier than trying to do it in something like corn.
So can you tell us what's going on in some of these pictures over here? What's going on in the upper left?
Oh, sure. So that was from upper left. That was from Halloween this year. We participated in the BTI Halloween contest, and we're actually what's called a restriction enzyme digest. And that means an enzyme is just a protein that's used to cut DNA, and that's used a lot for molecular biology work. So this is a DNA sequence. I am the enzyme, hence the hedge trimmers there. And so I'm cutting the DNA in the exact location where it needs to be cut. And then on the right, that's some of the work that we do. That's plant tissue culture work. And the plant tissue culture is the foundation for doing our genetic engineering and our gene editing work by CRISPR. The bottom left, that's Esperanza in the front and McKenna in the back. They put in our Physalis plants, our ground cherry, goldenberry, and tomatillo plants into the field in Ithaca, not far from BTI.
And you mentioned CRISPR and using molecular techniques to improve crops. Could you expand on that a little bit?
Well, there's traditional plant breeding that's the foundation for improving crops, but then there are also these techniques, CRISPR, gene editing, and genetic engineering that are used as tools to understand the function of genes, but also helps in crop improvement as well. And they're just techniques that we can use to modify the DNA. And CRISPR is the new technology on the scene, and it's very specific. And unlike genetic engineering, where you sometimes introduce new plant genes, new DNA, with CRISPR, the advantage is that you're tweaking the DNA of the plant species itself.
So you can also use that to find the functions of genes as well, right? So then breeders could, instead of just saying, hey, this one's tall, this one's tastes better, let's put them together and maybe we'll get one that's tall and tastes better. More specific when you look at the seeds, right?
Right. It's a really precise way of being able to narrow down which genes are involved in which characteristics, whether that be how the plant grows or how it tastes, nutritional value as well.
Okay, and how was your lab adjusting to the COVID-19?
Yeah, well, I'm not gonna lie, it's been stressful, and we had a plan in place. I have a great team, I have really good group, and we had a plan in place. We had a backup plan in place, both for the research and keeping people safe. We were had to go to reduced staffing and have just essential personnel be part of the lab. So we were able to maintain material, but not start any new experiments. So just like other groups, we have lost progress, but again, we were fortunate that we were able to maintain a material that we had and didn't lose it. And now that we're gearing back and reactivating research, we're able to start some new experiments that are part of our projects.
Great, that's good to hear.
Yeah.
And keep things moving. So my next question is about Physalis. You mentioned Physalis a couple times. Could you tell us more about that and what exact traits you're trying to improve?
Sure. So as you can see in my background, I am surrounded by Physalis. It's ground cherry and goldenberry. I have a real connection with my work. I even have a mug here. I don't know if you can see.
Good. Kind of lost in the virtual background.
Lost, yeah. Oh, there it is. There, Physalis. So it's the Physalis are related to tomato and potato and eggplant and pepper. They're part of a family called the Solanaceae, and the Solanaceae is made up of 3,000 different species that have all sorts of different uses, again, like for food crops like potato and tomato, medicinal crops, and also ornamentals like petunia. And the Physalis I have right here that we're working with are ground cherry and goldenberry, but the Physalis people might be most familiar with their tomatillo. So I have two here. You can see that right there. The one on, that's my left is, I'm not getting this too good
It's
here with
disappearing.
the camera.
Maybe if you hold it down.
Hold it down. Oh, okay, yeah,
There you
that
go.
works. All right, so the one here, that's ground cherry, and this is goldenberry, and you can see there's a difference in size. The ground cherry is smaller. They're covered by this husk, and there's different kind of theories on what the husk might do. It protects them from insects, from disease. There is also, kind of and how they evolved. It might have been some mechanism for them to be blown easily by the wind and spread the seeds, or to float on water.
Mm-hmm.
And so they're different in size, but they're this cute. I'm going to open these up now. There's cute little brown fruit. You can hear the husk is pretty crispy right now. You can see there's a difference in the fruit size. The ground cherry here is smaller. The goldenberry is larger. But there are also differences in flavor. The goldenberry actually has a more complex flavor. It's sort of this tart sweet mix of flavors, and some of them have undertones of mango or coconut and apricot.
Yummy.
Yeah, they are. And I hadn't had ground cherry or goldenberry before I started working on them. So this was, for our research. So this was really exciting for me and fun to be able to try something new. And then this is the ground cherry is overall a sweet flavor, not as complex a flavor as goldenberry, but we do have one that has a pineapple flavor that seems to be, a favorite of a lot of people.
Yeah, I like that one a lot.
Yeah, so we did last year we did a consumer preference study with Cornell Sensory Lab to have people do taste tests and both the ground cherry and goldenberry and give us some feedback. So it was really fun to see people's comments about their likes and dislikes. And the one thing they do have in common is that they have a very wild, sprawling growth habit, which is difficult, especially like I have a small garden. It's difficult to find space to grow them. But you can imagine, here, this Mike just put up this slide. You can see in the lower right-hand corner there, that's one of the goldenberry plants. So we have to do a trellising system to keep them upright. They'll just continue to grow. I mean, if you grow tomatoes and you grow these indeterminate type tomatoes, they're the same way. They'll just keep growing through the season and grow till frost and produce fruit until frost. And the goldenberry tends to grow more upright and huge. Our plants are like 6 to 7 feet tall, but the ground cherries are more sprawling. So that's their similarities in their growth habit. One of the differences is that ground cherry drops to the ground, and that's why it's called ground cherry, and it drops to the ground not even when it's ripe. It could be at different stages of ripening, even unripe fruit drops. And the reason for that drop is there's the connection between the fruit and the plant. There's this little area that looks like an elbow. It's a joint, and the fruit just breaks at that joint. And we're trying to understand why that happens. And we're using what I mentioned, molecular biology. We're using that to try to understand, and CRISPR as well, to understand what genes are involved in that fruit drop, because we're looking at this not just for our own research, but for potential as a specialty fruit crop in the US. And that dropping to the ground makes it really difficult to harvest. And not only would it make it difficult to harvest, but you have the chances of having more foodborne illnesses that could result from eating it, because they're on the ground, you're gathering up from the ground, and they're dirty. And even though they have the husk, you have to open them up and you can get the dirt on the fruit as well. The ground cherry makes more fruit than the goldenberry does. But that's another thing that we're looking at is productivity. And in this climate, and up here in upstate New York, the ground cherry, they'll produce fruit just like tomatoes. So they'll make fruit from the time tomatoes start to ripen all the way through frost. The goldenberries need a longer growing season. So we recommend that if people are growing the goldenberry, that they start them from seed much earlier than they would the ground cherries.
You mentioned that the ground cherries fall to the ground. I can see that picture of Brandon with a vacuum getting them off. Special vacuum cleaner? Is that a regular vacuum cleaner?
So you can see all the fruit drop there on the bench, and we used to use a broom and sweep it up, and a dustpan, and that took forever. And then we started using a regular shop vac, and we used to vacuum them up with that, but that was difficult to maneuver through the greenhouse. And then you'd have to dump it. You'd need two people to dump the shop vac. So I said to Brandon, I said, hey, you know, I wonder if there's a backpack shop vac. And if there isn't one, we should invent it. So he did some searching online and he found this backpack shop vac, which is just great. It really made it much easier to vacuum up the fruit and easy to maneuver through the greenhouse, and it could dump it. And I teased him because I told him he looked like a Ghostbuster, you know, with the backpack on him.
Yeah, a little bit.
Yeah. So, but you could imagine for a farmer to take this and have it large scale. It's really tough to harvest all that drop fruit.
Yeah, so I still don't see either of these in the grocery stores very much. Are those the main reasons why, the sprawl and the dropping of why farmers don't want to grow them in large quantities?
Yeah, you'll find you'll find ground cherries at farmers markets, but not a huge number of farmers grow them. And it's the difficulty with the sprawling growth habit. It takes up a lot of space and the drop fruit, but you will find them. And then golden berries are grown. The major exporter of golden berries are in Colombia, growers in Colombia. There are some from Ecuador, but you'll find those in the grocery store. Sometimes you'll find them in a husk, and sometimes they'll be out of the husk, but they're sporadic. You don't find them all the time. And it really didn't have golden berries coming into the US until maybe 4 to 5 years ago, because there was this fruit fly that was associated with the plants grown in Colombia. And the US Department of Agriculture really wanted verification and confirmation, made sure all sorts of practices were in place to prevent those fruit flies from coming into the US. But you occasionally will find the golden berries in the greenhouse. So our hope is that one day that farmers will adopt these in the US and grow both the ground cherry and the golden berry on a larger scale, but they do need improvement to make that practical.
Okay, that makes sense. And I have read that they have really good nutrition properties. Is that as well? I mean, could they enhance the diet in any way?
They do. That's another reason that we're looking at these as specialty food crops, because they really help diversify diets, and they have good nutrition level. They're good vitamin C. Vitamin C content is really good. Beta carotene, which our bodies convert into vitamin A. They've got a good amount of beta carotene, especially the golden berries, and you can tell by that color, the difference in color. Antioxidants. Something called plant sterols, which are plant oils. And what we've learned recently through some studies is they've got a good level of folic acid. And that's going to be one of Savannah, I mentioned Savannah Dale, a graduate student, that's going to be one of her projects to look at folic acid in these fruit. And what we've seen in the preliminary data that we've gotten, and we're going to follow up on more of this, it looks like if you eat 10 golden berries, you can get the required daily amount of folate.
Wow.
For an adult. So yeah, so it's
Could do that easily.
Yeah, I know.
So yummy.
It's eating too much, right?
And you mentioned that the Fishless could be used as kind of a model for other orphan crops. Are you working on any others, and how could these lessons translate to those other orphan crops?
Yeah, so what we wanted to do with our our research, both in the kind of our experiments, but also in looking at as a practical application to produce these as fruit, is to use it as a model for taking something, these plant species. That's the reason we chose this for our work, that are underutilized, and there's been little, if any, improvement that's been done to the ground cherry and golden berry. So it can serve as a model for the types of characteristics and the types of genes that would be important to work with in order to take other underutilized species and improve those to be able to be grown and be a source of food for people. Some other underutilized species, it's the same problems. It's this wild sprawling growth habit. Fruit or vegetables are dropped to the ground, small fruit, not a very high yield, but there are a lot of underutilized species out there. With some improvement, they could really be a viable source of food for people.
Very cool.
Mm-hmm.
Yeah, that sounds
So
great.
yeah, so your question about whether we have, so yeah, we're working on one called goji berry, which it's one of the things with goji berry that we're looking at is to make it more viable, is it takes about 3 years to go from seed to fruit.
Oh,
So
wow.
we want to look at, making that 1 year, and it's got a similar problem with the growth habit. It's really tall, make that more compact, but we're looking at other species as well. And goji berry is also within the Solanaceae family with tomato and the Physalis, but we have plans for other plant species as well. So it's been a
Okay.
lot of fun.
Very cool. And so my next question for you is about the your community science project, the Physalis Improvement Project. So what could you tell us a little bit about that and how that came about and what you're...
Yeah. So, so this community science project is my favorite project of my whole career. Don't tell my other projects that, but I love them. But this one is my favorite and has been most fun. And so the way it came about is we started working with, so we've been working with tomato, and we felt like the genes and the characteristics that we were looking at in tomato could be transferred to something that was, more distantly related but needed some improvement. And we chose the Fishless, we chose ground cherry and goldenberry. And as we worked with this, my collaborator Zach Lippman and I felt that there was some potential for these as specialty fruit crops. But I wanted to make sure that early in the process of improving the goldenberry and ground cherry, that we got feedback from farmers and home gardeners and consumers early on about what they felt the improvements needed to be, so that we could really be smart about what we were doing, and not guessing, but sort of using our own vision for what we thought was needed. So I thought really having that buy-in and that feedback from people was important. So what I wanted to do was get seeds of the ground cherry and goldenberry. We had 3 different types of each, 2 farmers and home gardeners. And we, but I needed to get the funding. So I really was like, I want to do this. How am I going to get funding? And as a faculty member, I spend a good amount of my time writing grant proposals to find funding for our research. So there's a local foundation in Ithaca called the Triad Foundation, and they provide funds to the Boyce Thompson Institute to help fill in those gaps where that can't be filled by other agencies like the National Science Foundation or the USDA. But it also, these foundation money can also give us preliminary data to go after grant proposals and additional funding from larger agencies. So I wrote a proposal to the Triad Foundation for this community science project. And I was so thrilled. I still get goosebumps when I think about it because it got funded, and it was really something I wanted to do. So I got the funding, and we stayed small that first year because it was not something I had done before. So we stayed within New York State, and we had farmers through different areas, different counties within New York. We also had some home gardeners involved, and also some organizations like the Ithaca Children's Garden, Keegan Nature Center, and Cornell Botanic Garden. As part of that, we gave them forms to fill in for feedback, like what did you think about the plant height? What about the flavor? Any problems with insects or diseases? And it was amazing because we got such good feedback, and it was, we didn't have money to pay the farmers to grow these for us, and we just had them adopt them into their systems, and they did a fantastic job with growing this material and giving us the really valuable feedback both from farmers and home gardeners about what the problems are with these plants that helped guide our work for improvement. And my favorite part of this is we got kids involved. Kids really enjoyed growing them and eating them. We did different events. And I was thrilled to see how much the kids liked them. Some of the kids even came back for more at some of the events. They enjoyed them so much. So I knew we were onto something. If kids like them, if you can make kids happy with something, then you're onto something good. The second year, so we got really good information on differences in insects and all sorts of other characteristics. So the second year, I used that information, that data that we got from that first community science project that was funded by the Triad, and I submitted a proposal to the National Science Foundation. and they funded it. So we're under funding with the National Science Foundation, and we've grown from almost 40 participants our first year and our second year, and now we're up to 700 participants in 43 different states, and we're focusing. Yeah, it is. It's amazing, and it's great. And part of that was this article that was in. I was interviewed by a writer for this article in the LA Times, both the online and the paper version. And it was really incredible, the amount of requests we got. They just started pouring in, and I think we were up to 1,400 some requests. I know, it was wow. Amazing. I loved it, but the reality is we have to do seed increases, and that means putting the plants in the greenhouse and growing up the fruit and taking the seeds out. And we only had enough seed to have 700 participants, but no, yes, I'm not complaining. It was 700, was great. I just felt bad we couldn't, give seed to all 1,400 people who requested them. So we're in 43 different states, and I was really happy. My goal was to get people in Hawaii and Alaska, and we did. So
looks like you did.
Yeah, we sure did. And this map here in the slides, each of those blue bubbles shows where the participants are. Who are participating. And this year we're focusing on this insect called the three-line potato beetle. And we learned so before we started our community science that first year, we had only grown the plants in the greenhouse. And by getting the plants out in the field and getting them to farmers and home gardeners, we learned about this three-line potato beetle, which actually prefers ground cherry and goldenberry over potatoes. So my first year, I had the most beautiful crop of potatoes in my garden because they all went to the ground cherry or goldenberry. They're really trap plants. But we noticed something interesting that first year, and that's why this year we want to focus on that in these different regions, these 43 different regions, is that the three-line potato beetle, the adults would visit the ground cherry and they would visit the goldenberry. They would lay eggs on both, but they only developed into larvae, like the little worms that come from the eggs, develop from the eggs. They would only develop into larvae on the goldenberry. If any did develop on the ground cherry, they stayed very small or died. So, all this information and data that we've gotten from people about this has now gone into this year, and we're using iNaturalist. We have a group in iNaturalist where people are helping us track the three-line potato infestation, but also other insects as well. We're interested in pollinators and any sort of other pests there might be or diseases. But that information, I put into a proposal with the USDA to be able to take this a step, look at this a bit more, like what's the mechanism, what's involved, what's causing this difference in the insect behavior. And again, using the ground cherry and goldenberry as a model, it'll be, if we're looking at improvement, that's important for ground cherry and goldenberry, that we learn how to manage the insects, but I'm hoping that we can use this as a model in other plant species as well that might have insect problems.
So that information about the three-line potato beetle, that came from the community science?
It came from our community science, yeah.
Oh, that's
and our
fantastic.
and that's really, and we need that information to put in that type of information, that preliminary data to go submit grant proposals. When I was a grad student, I thought, oh, you have an idea, you write a grant proposal, you submit it, but no, you need to have preliminary data to show, why you want to look at something and how you're going to look at it. But that was all from our community science program.
Yeah, well, that's fantastic, because there are data that show that these kinds of community science programs, getting public engagement, helps increase awareness of environmental issues and food, preserving biodiversity, all kinds of benefits for society at large, and so this is neat. I would imagine that people would be pretty happy to hear that they are helping, potentially fighting off this pest.
Yeah, I really do appreciate it. It's, like I said, it's been a great experience for me. I really appreciate everyone's wanting to be part of the project and putting in the time. And we have a Twitter account called @Physalis Project. And people are posting their pictures of how their plants are growing and the progress, and we're getting photos and information through iNaturalist. And it's been, like I said, it's my favorite project of my career, and it's because of the community scientists that have made it that way. But so I think it's really important for scientists, for researchers, to try to engage with the public and do some either community science or talk about what they're doing. I think the simple act of people meeting a scientist, it's especially kids, to see that we're normal, or we're okay people. We have interest outside of our work. We're passionate about what we do. We want to do good. I think that really helps start that communication. And if scientists aren't comfortable doing that, because there are people and organizations that help with that science communication. So I really recommend that people try to get out and talk about their work and engage with the communities.
Okay, that's great. So I guess my last question before we go to the Q&A is it possible for people to join this project at this point, or what's in store for the future of the Physalis Improvement Project?
Yeah, so I would love to continue, but the reality of our work is that I need additional funding, and one of the kind of fallout problems with the COVID is that a number of funding agencies have postponed calls for proposals until 2021. So while I would love to continue and have people participate, it's really uncertain at this point what we're going to be able to do until maybe 2021. I hope we can. I hope we can get the funding, and so that's where I'm spending my time right now, is trying to get some ideas, because a couple of the agencies that I thought I could submit a proposal to have postponed till 2021. So
that's too bad. This is really, pandemic's really kind of thrown a monkey wrench in a lot of things.
Yeah, it has, and it's just, we're not alone. I just have to remember that we're not alone. It's others are experiencing the same. But maybe we'll be able to continue on some low level. I don't know, but I hope something comes through without kind of, sometimes something comes out of left field with I'm not even expecting it, and I hope that's the case. As was the Triad Foundation in the beginning, getting the funding from them. I didn't know where I was going to get the funding, and then that came up, and that was a really benefit for me.
So people at home should stay tuned. Hopefully there'll be some good news.
Mm-hmm. Yeah.
Okay, do we want to try some Q&A?
Sure.
Hey, Joyce,
Mike.
AJ.
Hey, Mike.
That was great. Thank you, Joyce.
Sure.
So we have a couple questions here. So the first one is, do you trellis both of the Fishless species, or I guess all of the Fishless species?
So we're trellising the goldenberry because it grows so upright, and actually in Colombia, the farmers trellis the plants as well. It's a little more difficult to trellis the ground cherries because they do stay so low and flat. We try to stake them up somewhat when we first put the plants out there. They're on the stake, but they do, they're a little tougher to trellis compared to the goldenberries.
And can they be eaten raw?
Oh, definitely. Yeah, they are fresh fruit for sure. And they are used for, especially in the countries that grow them, like Colombia, they're used for making juices and jams. You can also get dried goldenberries. They're really good. Amazon has them. If you go to Amazon, they have the dried goldenberries. I looked for them in stores around here, but I haven't found them, but I've gotten them from Amazon. And if you go to our website, we actually have some recipes for using ground cherries and goldenberries. And one of those recipes is a granola that uses the dried goldenberries. So, but people want, so they can be eaten raw, or you can use them in different dishes.
Okay. So in your research, have you considered potential consequences to ecosystems when genes are altered that affect the phenotype? So in other words, what interaction could occur with other species in a natural environment?
Yeah, so that's something that even, with traditional plant breeding, people have to be aware of, and making those improvements, making sure that there aren't any consequences that come of that. So even before something goes out into the field, we make sure we do all the verification at the DNA level that the gene that we're targeting is actually affected and not other characteristics. Well, it's not just like we modify these. We're doing the modification and then putting them out in the field or doing a product. They've got to go out in the field and be evaluated to make sure that they're still producing, the yield still looking good, and that we're not making them more susceptible to insect or diseases, or kind of affecting other plants. So that's all taken into consideration when we're doing that.
And just to clarify, you're mostly silencing genes, correct? You're not
right. So what we're
producing
doing
new genes
is
from other species, or
yeah, exactly. That's a good point. So what we're doing with the editing is just going in, and that's the beauty of CRISPR, is that we can go in and just tweak that DNA itself, and we're making a cut in that DNA. It's kind of like a roadblock. If you're on the highway and you come across an area, it's a roadblock and you just can't go through, or you have to go a different way. And that's what we're doing. We're creating that roadblock, which is affecting the expression of that gene, so the function of that gene, so that it causes that change in characteristic, like plant growth habit. We know that there's a natural chance mutation that happened in tomato back in the 1920s. So if you've grown tomatoes, some of them can be really long and lanky and just continue to sprawl, but there are other tomatoes that are more compact and they're easier to grow, like in containers or in a small garden. And that's all because of a natural chance mutation in the DNA of a tomato plant that was observed back in the 1920s in the field. And researchers started to study, what is that gene? And it was just a cut in the DNA that prevented the expression and the function that took the long, lanky tomato plants to these smaller, compact plants. And that's exactly our model for the ground cherry and goldenberry. They're related to tomato, so that information on that tomato gene gave us the starting point for our work with CRISPR in the ground cherry and goldenberry. And we're creating that same chance mutation very precisely in those plants. And we have got more compact plants by just making that deletion or that change in that one gene.
So this is a three-part question. Are the Fishless crossings compatible with other species? How do the hybrids behave? And are the plants easily cloned?
Okay, so the first part was crossing. So even within ground cherry and goldenberry, we've tried crossing because the goldenberry does not drop its fruit, but the ground cherry does. So the first thing we thought was, oh, well, we'll do crosses, and then we'll evaluate all those plants that come from those crosses from the seed and look for ones that don't drop the fruit. We could not get the crosses to work. So that's why gene editing might be one approach to help with that fruit drop. Other characteristics, we might not have to use gene editing. We could just, within ground cherry or within goldenberry, we can do those crosses, but even trying to cross within those two, we weren't successful at getting them to work. As far as are they easy to clone, they're mainly done from seed. We have taken cuttings and rooted them in soil, so they root fairly easily, just like tomatoes would if you took a cutting and rooted those. And there was a second question, I wasn't sure.
Yeah, so it says, how do hybrids behave?
So, right, so if you make a hybrid with any species, what can happen is that, so you're taking in plant breeding, you're taking two different plants and you're crossing them to make something better. But what can often happen with hybrids is when you plant out those seeds, you don't necessarily get back what you started with. You're getting back the two parents. So each of those parents might have good characteristics that you want. And when you cross them, you get the best of both worlds in one plant. But when you try to grow those seeds again, you get something that's completely different. So the whole premise of hybrids is to be able to get something better, whether that's better yield, better growth habit, better disease resistance, those types of characteristics. Okay.
So like we have two questions here in a row from some citizen scientists, and they're pretty much the same question. So is it possible for home gardeners to save ground cherry seeds for plant next year? And then should we be saving our seeds?
Oh, that's a great idea. It's a great suggestion. Yes, you can save the seeds. I mean, that's how we generate the seeds for the Citizen Science Project, the Community Science Project, which it's called Community Science now, is we grow the plants in the greenhouse and we collect the fruit and we have the seeds, and that's how we generated all the seeds for the project. So yeah, it's a great idea for the home gardeners to collect their seeds, their fruit, and take the seeds out.
Great.
Can I ask a quick follow-up on that, Joyce? Do the seeds need any treatment before planting them? Like, do you need to put them in the freezer for a couple weeks or anything like that, like you do with strawberries and other?
Yeah, no, that's the really nice thing about these is, as you mentioned, some seeds you have to either put them in the freezer for a while, or even some seeds you have to store them for a longer period of time because they have something called a dormancy and they won't germinate, or you have to nick the seed coat or do something special. But these are just like tomato seeds. You extract the seed, take the seeds out and let them dry, and then you'll have seeds for next year.
Cool.
Yeah, they're easy.
So the next question is, do you know what could be explaining the difference in insect development in the two different Physalis? Have you checked differences in different specialized metabolites on them?
Yeah, that's exactly what we're doing right now. So I love that question because that is the premise for this proposal that we just submitted. So insect interaction can be physical, right? It could be hairy leaves, it could be something about the leaf structure, but it also can be these specialized metabolites that are produced by the plants. And Physalis, what we're doing now is doing this kind of broad screen to look at the differences in the metabolites or the compounds between ground cherry and goldenberry to get this view of what the differences might be. One of the metabolites that we're focusing on right now, but again, we're keeping an open mind to look at others, are called withanolides. And they're a steroidal compound. And we're seeing differences in the types and what they call the profiles between ground cherry and goldenberry, which could explain the differences. But we need additional funding to be able to take that the next step further to look at that. But there could be other metabolites as well. But the main ones right now are these withanolides, and withanolides are not produced by all plant species. It seems to be just a small number, around 7 or so, and within the Solanaceae and the Physalis do produce good amounts of withanolides.
Next question, are there known medicinal properties of Physalis?
Yes, the Physalis have been used for long decades and centuries for the medicinal properties. Part of it is these withanolides I just mentioned. They've been used for different ailments like anti-inflammatory, where a lot of the research is in anti-cancer compounds, to be using those. They've shown good inhibitory effects on cancer cell lines, and there's quite a bit of research at looking at not only just from ground cherry and goldenberry, but other Physalis as well for the medicinal properties.
Can the different species be cross-pollinated, or does tomatillo only pollinate with other tomatillo?
So, interesting fact about tomatillo, even some tomatillo don't cross with each other. And some of them have what's called for tomatillo is a self-incompatibility. We actually did a blog post, if anybody's interested, about self-incompatibility, I don't know, maybe a year or two ago, that even the pollen from that same plant won't produce fruit. So within tomatillo, it's even difficult. So what you sometimes have to do is plant not just one tomatillo, you have to plant two different, same tomatillo, but two different plants so that they could cross-pollinate with each other.
Here's a great question. What ways can citizen scientists get involved in the Physalis Improvement Project if they're not currently growing seeds directly from your trials, but are growing a wide range of sometimes less common Physalis species?
Oh, I would love to hear about that, because that's something that we're going to start expanding into other species. And one of the additions that we did to our ground cherry and goldenberry project this year is that in addition to people growing them and giving, taking pictures of the insects and the pollinators, was what we call Physalis in the wild, or Physalis other than the ground cherry and goldenberry to let us know what they're growing or what they're finding in the wild. And that could be done through iNaturalist and our group on iNaturalist. And information for being able to be part of that is on our blog and on our webpage for the Physalis Improvement Project.
How many other wild Physalis species are there? I mean, are there like wild ground cherries and things in the?
Yeah, you know, ground cherries, there was a great article recently. I can't remember if I can't remember the journal, the magazine, about ground cherries being part of, you know, the Native American diet, and these species that are, you know, that had their origins in North America. And so they were just growing wild there for ground cherry. And there are other Physalis species that are, you know, so different plants have different origins in different places. Like the goldenberry has more of an origin in South America, whereas the ground cherry is more North America, and was part of the cuisine, you know, a long time ago. And these chefs are starting to try to bring it back, some of these different species, and ground cherry is one of those.
So we have a question from Canada here. First off, they say, thank you for doing this and for your great website. So, great website. So the first part of the question is whether goldenberries can be grown in the ground without killing over winter. And the second question is there a natural way to eliminate the potato bugs that might infect the plants?
So, yeah, so we had pretty short growing season here as well. And the plants, I've kept my plants in my garden, like my goldenberry especially, they just don't make it through the winter. There are some farmers who try to extend the season of plants by using what's called high tunnels, so they can get plants started earlier and keep them later. But our ground cherries and goldenberries just don't make it through the winter. One interesting observation we did make is that we're growing like goldenberries from different sources. We have goldenberries from India, South Africa, Colombia. And in our field last year, we noticed that the plants from Colombia actually lasted longer in the field and continued to produce fruit even when it got colder. So there are differences in germplasm and sources that people can use depending on their growing environment. And so that's you know, one way to be able to extend the season for that. What was the other part of the question?
I think, let's see. Oh, and then is there a natural way to eliminate the potato bugs that affect plants? So a natural way to.
Okay, so one of the ways we've been doing it, and Esperanza, who's in charge of the Citizens Science Project, actually squishes them. She takes them and she goes in and she's done hundreds of them trying to keep them off the plants, and she's done a great job. She should get a bonus for that. And anyway, there are also, I haven't tried it myself in my small garden. I just squish the adults or the larvae or the eggs when I see them. Some people have recommended trying like a mix of soapy water with garlic in it, or I've also heard of, you know, kind of some other homemade type remedies that people could use. But other than that, I'm not aware of it. I know there might be some things that organic farmers might use, but I'm not familiar with those. But maybe a quick look on the website, on the internet, would be able to get that information.
Yeah.
Maybe we have time for one last question.
Yep, we have one more here. Can they grow inside the house?
I haven't tried it myself. I think if you had a really bright, sunny spot, that they might be able to make it. You might have to cut them back to be smaller. I think it's worth a try to grow them in the house. But again, they would need a really highlight area to be able to grow. They love the sun. They also they don't like to be wet. They tend to rot if they're too wet, so you need well drainage. But sure, I think you could give it a try. You might have to trim them back, but if somebody tries it, I'd love to know if they got it to work.
All right, great. Well, thank you again, Joyce, for taking time to chat with us all today.
Mm-hmm.
Much appreciated.
And thank you for giving me the opportunity to talk about the project. And thank you to everyone for your questions and for tuning in. I've really enjoyed it.
Yeah, thank you to everyone who joined us for this new series of virtual chats. And you can read more about Joyce's Community Science Project, the Fishless Project, and many other neat stories about BTI science in our 2019 annual report, which you can find online at btiscience.org/annual report. Looks like Mike just popped the URL in the chat box there. And this year, it's all digital. We didn't print one. So it is all digital, and there are 3 different views. There's like a single view, a double page view, which is most similar to reading like a physical copy. And there's also like a reading view, which is like reading a website article, like the HTML article. And there are many links to videos in there, so it's very interactive. So it's pretty neat. So we hope you check it out. And we also invite you to join us for BTI's next Breaking Ground discussion series, which will feature a similar interactive chat like this with Dr. Fay and Dr. Giovanoni on Thursday, July 30th at 12 noon. And we'll be talking about their work on the tomato pin genome, which has a lot of neat stuff on with it. And you can go to btiscience.org for more information and to register. And it looks like Mike just popped that URL in there as well. And just like to reiterate that Boyce Thompson Institute is an independent nonprofit research institute. And we operate in part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give or email our development team at development@btiscience.org. Looks like Mike put those in there as well. So thank you, Mike. And thank you all. Thank you all for your interest and your support of BTI. And I hope you have a wonderful rest of your day and be well. Bye all. Bye, Joyce.
Bye. Bye.